Thank you very much for the opportunity to present my results of our Gnatwork project to you today. This project was a collaboration between myself at Pirbright and Dr. Godwin Kwatrie-Nweko at the University of Cape Coast in Ghana. And Godwin's research assistant, Priscilla Ankama-Opare, has also been highly involved with this project and has been instrumental to its success. I will talk briefly about the background to the project, which is partly based on work that Godwin carried out for his PhD at Lancaster University. I'll describe the methods of our project, our findings, and the future work that has been generated as a result of this seed funding. So firstly, there are two main types of leishmaniasis in humans: visceral leishmaniasis that can be fatal, and cutaneous leishmaniasis, or CL, which causes potentially disfiguring scars. CL itself can take a number of different forms, but all are primarily caused by the protozoan parasite Leishmania. Over the last 20 years, cases of Leishmania have been reported from new regions, and additionally, new species of the parasite have been discovered. Autochthonous cases of human CL were first identified in Ghana in 1999, and numbers rapidly increased over the next few years. Human leishmaniasis is known to be spread by the bite of an infected phlebotomine sandfly, but here we will present a slightly different story. In 2015, Godwin identified the causative agent of CL in Ghana as a new species within the Leishmania inrietii complex, which has been variously named GH5. The Inrietii complex, which has now been classed as a new subgenus, Mundinia, includes species from Thailand, the Caribbean, and Australia. Very few phlebotomine sandflies have been collected from endemic foci in Ghana, and indeed GH5 replicates and disseminates successfully in experimentally infected Culicoides sonorensis in the lab. Notably, it fails to replicate in a permissive phlebotomine sandfly vector. It has therefore been proposed that there may be a novel transmission cycle of Leishmania in Ghana that utilises Culicoides midges instead of the traditional sandfly vectors. Previous trapping in Ghana, of which there has not been very much, has shown that a single species of Culicoides was biting humans, yet this species did not show up in CDC light traps. However, the use of CO2 as an attractant has been proven to increase the diversity and abundance of Culicoides and other Diptera caught in light traps. So that is the story so far, and what led us to the following objectives for the Gnatwork project. Firstly, to collect Culicoides and sandflies from a Leishmania endemic region of Ghana using CO2-baited light traps and human landing catchers. Secondly, to identify collected Culicoides to species level to incriminate potential vectors of human CL. And finally, to validate a CO2-baited CDC trap against explicit estimates of human biting. Our field site was a small community in the southeast of Ghana that's been affected by human CL. The village is called Dodome-Awiasu. We selected 4 sites within the village: the water pump, a courtyard surrounded by homes, the front yard of a house, and the local church. The health centre, or nurses station, is marked here on the map as well, as we had a CDC trap running here throughout the duration of the study. These are 3 photos of the 4 sites that we used. So on the top left is the front garden of somebody's home. The middle picture is the water pump, and the bottom left picture is the road next to the church. Collections were carried out by 3 human volunteers and a CO2-baited light trap that acted as a 4th pseudo-volunteer. We conducted a total of 15 nights collections, and each collection was 2 hours long. Each human or trap spent 25 minutes at each of those 4 sites within the village and then rotated, and rotations were planned a priori by a Latin square design. On humans, insects were collected using a filtered mouth aspirator and were then frozen and stored in 70% ethanol. And as I previously mentioned, we ran another CDC trap opportunistically at the nurses station. These are some photos from the collections. So on the top left, we have the CDC trap, and in the bottle at the bottom is fermenting yeast producing CO2 as the attractant. In the top right, Priscilla is carrying out a human landing catch next to the water pump. And unfortunately, there were a few bites where people were not quite quick enough with the aspirator. At this point, it's worth mentioning the impact of COVID on our project, as it was not insignificant. Our fieldwork was originally planned for March and April 2020, but this was delayed due to lockdowns everywhere. Ghana came out of lockdown in May, and it was possible to commence fieldwork immediately. So our fieldwork in the end ran from May to August. Unfortunately, this coincided with the wet season, which also means a higher risk of leishmania transmission. However, all our volunteers were happy to continue, for which we were very grateful, and signed consent forms to that effect. Lockdown in the UK prevented my second trip to Ghana to provide training and assistance with the identification of the Culicoides specimens. So in the end, Godwin and Priscilla posted their collections to me so I could identify them from my lockdown microscope at home. Whilst we were waiting during lockdown, I produced a key for the Culicoides of Ghana, those that are known to be there, plus those that were considered likely candidates to come up in our collections, being found in nearby countries and in equatorial regions of Africa. This key is not exhaustive, but will hopefully be a useful guide and is available on the Gnatwork website for anyone to use in the future. Okay, so on to our results. We collected 1,320 Culicoides from our human landing catches and our pseudo-human, of which we were able to identify 1,280. Culicoides grahami dominated both the human and CDC trap catches. This has previously been found elsewhere in Ghana, and it is a member of the subgenus Avaritia. We found 5 additional species, but in very low numbers. We collected 887 other dipterans, of which 18 were sandflies. Following our statistical analysis, we found that the CO2-baited trap collected significantly fewer Culicoides than the human volunteers, with humans collecting 4 to 6 times as many as the trap. There was no statistically significant difference between the 3 humans. In terms of the different sites, significantly more Culicoides were collected at the water pump and the church than at the 2 dwelling sites. We therefore propose Culicoides grahami as the putative vector of leishmania in this area. Unfortunately, we cannot recommend a CO2-baited CDC trap as an accurate proxy of human biting rates. But we can recommend that members of the community avoid using the water pump and hanging around the church during the hours of sunset to reduce their chances of getting bitten. So this is a summary of the main outputs from the project. Firstly, as I mentioned, we have the key for the Culicoides potentially found in Ghana available on the Gnatwork website. Research staff in Ghana are now proficient at setting up and using CDC light traps and carrying out human landing catches. We purchased and installed a new dissecting light microscope for use in the lab at UCC Ghana. And all the fieldwork equipment and CDC light traps used in the study remain the property of UCC for future insect studies. Additionally, Godwin visited Pirbright just before lockdown for basic training in Culicoides identification, networking, and he also gave a seminar to Pirbright during his visit with us. We've been successful in securing follow-up funding from BBSRC through Pirbright for a 10-month project, which is currently funding Priscilla. This has given us the opportunity to identify the remaining samples from our ad hoc trapping, to test Paris Culicoides for the presence of leishmania, and to carry out blood min analysis to confirm human biting and potentially identify reservoir hosts. All being well, in July, Priscilla will be travelling to Pirbright to carry out this work. At this point, I'd like to put in an unashamed plug for Ento 21, which is the Royal Entomological Society's biennial conference in August. Priscilla will be speaking at that and hopefully presenting some further results. So please do come along if you're able. Finally, we'll be conducting a workshop with residents of Dodome-Awiasu to share our findings with them. We're currently also working towards 2 larger grants, an MRC responsive mode application, which will be primarily to dissect field corculicoides to detect the different developmental stages of the parasite, and also to investigate the natural reservoir of leishmania. Secondly, we are looking at a PhD application to expand the fieldwork that we've already carried out in the Gnatwork project across a wider geographical area, and to also look at the epidemiology of leishmania in endemic foci. So finally, it remains for me to thank the Gnatwork very much for the funding opportunity, and also thank you to everyone who voted in the community call. Godwin and Priscilla have been absolutely fantastic to work with, so big thanks to them, and also the villagers of Dodome-Awiasu who welcomed us with open arms and have been really supportive of the project. Thank you finally to Emma, Alex, Simon, Jess and Chris Sanders for their support throughout the project. Thank you.